Lou, are you actually in the gardens today?
Yeah, I've been here most of the time. It's actually quite sunny today up here.
Really?
Sunny in Bakersfield.
Yeah, I can imagine, John.
It didn't start sunny here.
Go ahead, Lou.
You tell me when.
Go ahead. We're all good.
You want me to start?
Please.
Okay. Good afternoon. My name is Louis Feldman, and I'm the Executive Director of the UC Botanical Garden and also Professor in the Department of Plant and Microbial Biology. Today, the Botanical Garden, along with the Innovative Genome Institute and the Department of Plant and Microbial Biology, are among the sponsors of today's event, and we are very proud to partner in this event. And despite the forecast of rain this weekend, which would make it difficult to come to the garden, I want to encourage you to come to the garden because there are a number of spectacular flowers here at this time, and you may even find some fungi, although it's kind of late in the season for fungi. I'm delighted to be spending part of this afternoon with you, and before I introduce our moderator, Iman Silvan, I want to mention that today's discussion is part of a series of events programming to honor the 30th anniversary of the Department of Plant and Microbial Biology. And this department has had a terrific contribution record, and it's one in which I think we can all be very proud of. We all look forward in the future, however, to seeing you in person, perhaps at a special anniversary event once the COVID-19 restrictions end. Today's moderator, Dr. Iman Silvan, received her PhD in 2018 working with one of our speakers today, Dr. John Taylor. Dr. Silvan has a record of great interest in indoor health and social justice, and completed her dissertation working with Dr. Taylor, trying to understand the impact of mold on low-income populations. After completing her PhD, Dr. Silvan went to work for the city of Berkeley, working with a city council member, Cheryl Davalia. And after that, she worked in the Innovative Genome Institute, was studying and working on COVID-19 testing. She has been a member of this unit for a period of time, and now she's one of the panelists in today's event. So Iman, I'm going to turn it over to you. Thank you.
Thanks, Lou, for that introduction. So I just want to mention that we'll be taking live questions from the webinar chat function or by YouTube Live. And this talk will also be recorded and available on YouTube shortly after we finish. So I would like to introduce our two panelists, John Taylor and Tom Bruns, who are renowned fungal biologists who have revolutionized the field of mycology by using PCR and genetic sequencing to identify fungi and establish their evolutionary relationships. Their seminal paper, Amplification and Direct Sequencing of Fungal Ribosomal RNA Genes for Phylogenetics, now has over 33,000 citations. I've had the pleasure of getting to know these professors during my time at Berkeley when they served as my faculty mentors. Tom was a member of my dissertation committee and is a famed expert in the field of fungal biology. He takes classes to field trips to Mendocino and Yosemite, and he's earned overwhelming praise from students at UC Berkeley who have honored him with an Outstanding Teacher Award. He is respected by colleagues across the campus and across the globe. Tom's current research includes post-fire saprobic fungal communities, mycorrhizal facilitation of invasive pines in the Southern Hemisphere, and longevity of ectomycorrhizal spore banks, and mycorrhizal facilitation of bristle cone pine expansion in the White Mountains. And John Taylor was my PhD advisor at Berkeley, where he was a professor for over 40 years. He and Tom Bruns have taught courses together for over 20 years. John's classes include field trips to wineries, cheese factories, and mushroom farms, and an epic overnight mushroom hunt with mycology courses from 5 Northern California campuses. That is an awesome trip. John has been the president of the Mycological Society of America and the International Mycological Association. His current research addresses the role of fungi when crop plants experience drought and the environmental reservoir of the fungus that causes the San Joaquin Valley fever. So welcome to you both. And just to begin, I want to just mention that we know the role of fungi in the environment is as decomposers. Fungi are able to break down biomass in plants and animals and release constituent nutrients back into the ecosystem. When fungi break down matter that's already dead, we call them saprobes. When fungi break down bodies that are still living, we call them pathogens. And within the last year, the experience of the COVID-19 pandemic has shown us how critical it is to study the ecology of pathogenic microbes. I hope we can come away from this period with a heightened sense of value for basic research in microbiology and the importance of scientific literacy for policymakers and the general public. So it's in an effort to share this information about microorganisms and in celebration of the anniversary of PMB that we delve into this discussion of the fascinating world of fungi. Now, the world of fungi is also our world. We have co-evolved with fungi. They're omnipresent in our ecosystems, in aquatic environments, in agriculture, in the air, in buildings, and even in our bodies. And there's so many natural history stories that I'd love to share with you, but let me turn it over to the experts so they can share some of the awesome features of fungal biology that intrigue them most. So, John, can you tell us some of your favorite stories of how fungi interact in modern society? I'm thinking industry, agriculture, cultural identity.
Well, I'll start out with I'll start out the same way Tom and I start out mycology courses. Most people think of fungi, they think of a mushroom. It's the first thing you think of. And then if they know more, they think of the drug penicillin, which they may remember was the first antibiotic, and it was made from a fungus, from a penicillium species. And then if they really know a lot, they will remember that yeast is a fungus, and yeast is certainly the most famous of all fungi, but I think maybe not recognized by many people that it is a fungus. And it's so famous industrially because all bread that rises uses yeast because yeast produces carbon dioxide. And all alcoholic beverages are made from yeast. And the alcoholic beverage industry is $1 trillion a year industry. And as Tom is fond of saying, all societies have developed an alcoholic beverage. Did you want to add something there, Tom?
I think we're going to go on to the ag side of it shortly. So no, I guess I would add the molecular biology side of it, that the other, in modern society, one of the big inputs that fungi have provided are model systems for understanding the basic mechanisms of cell function and communication and so on. And it's resulted in 3 Nobel Prizes, 2 Saccharomyces, essentially. So.
Very good point. And I would add a little biology. Yeast is a little sphere, and the food diffuses to yeast. So that's why it's really good. You can throw it in a vat of sugar, and it will produce ethanol and CO2. But a lot of the fungi that we'll talk about today grow as little tubes from their tips. And so they can invade the food supply, export enzymes, break it down, and import the small molecules, and then just grow forward into new food. So yeast is actually in some ways an atypical fungus for many of the fungi that we'll talk about today.
Yeah, I love the idea of fungi eating through their environment as they grow.
It's a great thing, for example, in a cheese, where the fungus may be eating through the curdled milk and converting it into a more palatable cheese. But it's a sad thing when it's in the wood that makes your house.
Yeah. Well, on the subject of housing, have you heard about fungi being used as packaging materials or for fashion or furniture?
Yes.
Yes.
Can you tell us a little bit about that biology or that industry?
So actually, Tom, I'll let Tom tell you about Philip Ross. He came to visit us.
Yeah, he came to visit us quite a few years ago now, and left some samples with us of some of the packing materials that you mentioned, but they use them structurally too, where basically they can make a mold and put in wood chips and then inoculate it with a fungal strain that then grows through it, and and solidifies it all, and then they bake that, and it's strong and firm and recyclable. And one of the newer things they've worked on is substitute for leather. That there's now a fungal leather-like material that's currently probably not competitive because of price, but will eventually get there.
So fungi have interesting applications for sustainable products. One of which that I'm really excited about is like alternative meats. So before Beyond Burger, before the Impossible, there was corn with a Q, Q-U-O-R-N, which is a mycelial product. What are some other ways that fungi have interacted in agriculture and food and cultural norms?
Well, a key element of the interaction of fungi and crop plants is little appreciated. And it turns out in the roots of plants, there are fungi that are neither decomposers nor parasites, but they're commensal symbionts with those plants. And in fact, 90% of the plants on Earth have fungi associated with the roots. And some of them are fungi that are associated with large trees. And I'll let Tom tell us about those first.
Yeah, so in our northern forests, our temperate forests, and in the far southern temperate forests, the trees are associated with what are called ectomycorrhizal fungi. That these are ones that often form large mushrooms and so on, and things that we collect and eat, like chanterelles and Aspergillus and matsutake, so on, are all examples of ectomycorrhizal fungi that are mutualistic with the trees. And these trees basically can't live without those fungi. That if you take the tree and move it to a part of the world where forests like that don't natively exist and the fungi aren't there, the trees cannot grow until the fungi get brought in. So pine, for example, did not initially grow in some areas of the southern hemisphere that they were taken to until these mycorrhizal fungi caught up with them. So it's really an obligate mutualism here, and it's an expensive one that the trees pay out 20 or more % of their net productivity, but it's a good tax in that without it they couldn't grow there at all, and with it they can grow to much larger sizes.
And the fungal networks below ground connect multiple trees, so you can move nutrients through an entire network. And then there's even some orchids that tap into that network and steal carbon from the trees and don't have to produce their own photosynthesis.
Right. Yeah, there's a whole set of different plants that have learned to parasitize this system, where they basically plug into it and sponge off it. In California, our best examples of this are things like snow plant and pine drops, and I forget the common name for Corella rhiza, the red orchid that's not
Coral root orchid.
Coral root orchid, yes.
Yeah, I think if there's one fact that amazes me the most about fungi, it's that no fungi, no pine trees, no oak trees. And we talk about lichens a lot, where a lichen is a combination of a very simple plant, an alga, and a fungus. And that's the big example of an obligate symbiosis. But to me, the ectomycorrhizal symbiosis with large trees is even more impressive. But there's a whole nother set of mycorrhizae, which is fungus root mycorrhizae. And these are little ones that you would never see. They don't make a mushroom. They're distantly related. But they've been with plants since the time plants came to land. And they're found in fossil plants that don't even make roots. So you could make the argument that plants evolved roots to provide an organ to interface with these mycorrhizal fungi. And they're in crop plants. And we just, my lab working with Peggy Lameau in our Department of Plant Microbial Biology, and also members of the department who are adjunct professors from the Plant Gene Expression Center down in Albany, a USDA facility. looked at sorghum as it underwent drought in the Central Valley, because it's a drought-resistant plant. And fortunately for us, a scientist at Cornell, Maria Harrison, had found that there are about 200 genes that sorghum turns on when it has one of these mycorrhizal fungi in its roots that aren't expressed when it doesn't. And it turned out that when we subjected the plants to drought, so we subjected them to drought until they flowered, that those genes were reduced in expression, and the abundance of the mycorrhizal fungi was reduced to the same extent. But the fungi weren't lost, they stayed there. And when water was added back, the plant gene expression came up and the abundance of the fungi came up. And that was the biggest coordinated response the plant made, which we as mycologists take as evidence that those fungi are extremely important to the plant and important to surviving drought.
Yeah. So to delve more into fungi and agriculture, which is actually how I got interested in mycology, I was first studying coffee agroecosystems. And then there's a coffee rust fungus, which has like decimated all of coffee production in Ceylon, who then switched to tea, and also banana leaf streak, which was a fungal disease that wiped out the banana republics, essentially. I'm really curious about this dual role of fungi in agricultural systems as pathogens, as killers of these crop plants, especially in monocultural production, but then also the providers of food. So can you talk a little bit more about this duality and nuance of fungi and ag?
I think it's kind of a human construct, this duality, because you can look in either of those compartments, the pathogen or the mutualist, and find both positive and negative interactions within them. So for example, a lot of fungal pathogens are actually beneficial to humans because they are pathogenic against things like insects and so on that we're trying to control. And so the fact that they're a pathogen alone doesn't necessarily make them negative unless they're a pathogen on what we're trying to grow for our food.
Yeah,
and,
I feel
likewise, even the fungi that make mycotoxins in our food and so poison it, those are some of the same fungi that we've exploited for antibiotics and medicines and so on.
Yeah, so perhaps it's just a matter
much
of
more nuanced on who's a positive impact and who's a negative one.
When you get into trouble in agriculture with fungi, is because you've grown vast acreage of one kind of plant, maybe even one clonal genotype of plant. And so if a fungus that has evolved to attack that plant, which usually did not come from the area that you're growing, it came from somewhere else on Earth, when that fungus reaches it, like probably coffee rust when it reached vast coffee plantations in what was then Ceylon, boom, it has a field day and the farmer is in huge trouble. So it's a problem of the way that we conduct agriculture.
Yeah. And a part of this question of nuance, maybe we could just kind of find ways to switch the narrative. So one of my favorite natural history stories of fungi is Wetlacoche, Ustilago, which is a fungus that parasitizes corn, and it creates essentially tumors on corn. And in Latin America, it's considered a culinary delicacy, and it's eaten in quesadillas and tacos. And so where there's a cultural relevance, this is not considered a disease. It's not a problem. But elsewhere, we're producing corn mostly for industrial products or not necessarily as food only. It's considered a parasite, and so we try to eradicate it. So I think finding a new position can switch the narrative about what is wrong, what is bad, what is positive and negative in fungi.
Absolutely. Tom and I, we teach a class, Fungi History and Society, and in that class we have made salsa with Wetlacoche.
Really?
You can sometimes get fresh ones at places like Monterey Market because they sort through the fresh corn, the and they throw, they put aside the ones that have the galls because people won't buy it. But you can also buy it canned at markets that have a Mexican theme.
I have to say it's an acquired taste primarily because it looks awful. If it's just gooey and dark and whatever, but it's lovely. It has a great flavor.
Yeah. So there are some very clear intersections between fungi and public health. We know that opportunistic pathogens are like the #1 killer of immunocompromised patients. So what this means is someone might contract an HIV virus, but ultimately die of a fungal infection. And when we say opportunistic pathogens, we mean fungi that are just kind of in the air, not explicitly trying to cause disease in a human, that then get incorporated into a body of someone with a weakened immune system, and then their growth kills them. So haphazard interactions in nature can be fatal when the immune system is not strong. And yet, like as you said, Tom or John, that penicillin was our first antibiotic produced by a fungus. So again, here's this like dual role of like saviors, friends, and foes in public health and medicine. So can you talk a little bit more about some interesting stories of fungi in the role of health?
Well, as you mentioned, fungi, well, if your immune system is severely compromised, so that would be HIV before the highly aggressive antiviral therapy, or if you're suppressing your immune system to get an organ transplant or for cancer therapy, then you're at risk, because our immune system is really good and it keeps fungi out of us. But as you said, there are fungi that can grow in your lungs, for example, when your immune system is suppressed. So in fact, the defining disease for the AIDS epidemic was pneumocystis pneumonia, which is caused by a fungus that is commensal in the lungs of, we were talking about this today with my postdoc, 20% of the population is the estimate. But it can be spread by air. So if you didn't have it if you had it in your lungs and you become immune suppressed, it can kill you. Or if you become immune suppressed and somebody breathes on you and you get those spores, it could grow on you and kill you. So that's a real problem. With the antiviral therapy, those diseases have pretty much gone away in the United States. But if you go to a part of the world where the antiviral therapy is not widespread, for example, Africa, then the fungus Cryptococcus, which also causes a pneumonia, is either the principal cause of death, or it's in a race with tuberculosis as the principal cause of death of AIDS victims in Africa.
You want to mention Fimigatus too, Aspergillus?
Yeah, so there's this is a fungus, Aspergillus Fimigatus. Okay, an interesting thing is not many fungi can grow at body temperature. So that rules out a lot of fungi. It rules out almost all fungi. There may be 100 species out of 10 million that can grow at body temperature, but Aspergillus Fimigatus is one of them. It grows in compost piles, which are hot because of microbial activity. And so that can cause a pneumonia in people whose immune systems are suppressed. And your immune system has to be really suppressed. More than HIV would be the suppression for an organ transplant or cancer therapy. And an interesting and tragic problem that's just arisen is that there are antifungal drugs that are developed for medicine, and the azoles, itraconazole, ketoconazole, there's a whole fluconazole, there's a whole family of them, were developed to fight fungal infections in people. But it turns out they also fight fungal infections in agriculture. And there's a lot more use in agriculture and a lot more profit by selling them in agriculture. And now we have developed azole-resistant Aspergillus Fimigatus. And it's a problem in the ICUs in Europe. It's not a big problem in the United States yet, but it almost certainly will become one.
Yeah, President Obama had a big initiative to reduce antibiotic use in agriculture. That was an aside. Tom, can you?
That's a really good point, because here's the problem. You're a company, you developed this antifungal drug for human use, and then you realize you could make 4 or 5 times as much by selling it in agriculture. So there, you know, that's going to require some amazing social effort to stop that.
Mhm. Tom, can you share with us a little about how shifting climates and global change are impacting fungi and forest communities? And can you share with us a bit about your work with wildfire communities?
Okay, I guess those are related. First, I would say that like much about global change in the biological level, there's more that we don't know than we do know about it, and so presumably fungi are experiencing the same sorts of things that all other organisms are. That their habitats and so on are changing as a result of global change, and so they'll find themselves somewhere where they don't grow as well, or the spot where they could grow better is now distant from them, or something like that. So there's all of that going on. And then in terms of things that we can see, one of the most interesting, I think, is that the fruiting period of fungi has shifted. We see this in California, certainly, that a lot of the sort of peak fruiting of mushrooms in our area, for example, was always kind of mid-October through November, and it's really been pushed back almost a month now. That peak fruiting is really much later, because the rains come much later than they used to. And in England, a long-term study of fruiting showed that some fungi actually have 2 periods of fruiting now instead of 1, that they'll fruit both fall and spring instead of just one of the 2, because the optimal temperature rainfall hits at 2 different times of the year now. So it's a real mixed bag, like global changes for all organisms, and I'm not sure what the predictions are on it, but fungi will play out in that arena like all the other organisms.
And as we experience habitat loss, are we also experiencing the extinction of fungal species?
Possibly. And again, I think we know so much less about fungi in terms of their distribution and so on, and how, partly because many of them can wait out long periods of problems by hanging out as resistant spores in the soil for decades and so on. So they might have more resilience, at least some of them may have more resilience than other organisms in that realm. But you know, decades is still a short time frame when you're looking at a global level.
Yeah, I'm just thinking of Elsa Vingelis, who successfully put a fungus on the endangered species list.
I'd forgotten that.
Yeah, I was thinking that we needed Elsa here to tell us about
The
that.
red listed fungi, yeah.
Yeah, and a lot of those, I think that started in Northern Europe, and a lot of those fungi were fungi that decayed trees that have become rare because of overcutting of
Right, and that,
trees.
so it was basically habitat loss. I mean,
Yeah, it's habitat loss. There's a possible change in the distribution of a fungus that I'm studying now that you mentioned, the fungus that causes valley fever, and it's known. Behind me is a picture of Bakersfield, sort of the eastern edge of Bakersfield, and I'm actually in Bakersfield now sampling soil with my technician Lily Montoya and postdoc Rob Wagner. And it's predicted based on climate studies that the range of this fungus is going to move northward. And it's already, there are a few cases in eastern Alameda, eastern Contra Costa County, definitely in San Joaquin County. So it could move north, and there's good evidence, there's evidence of viable fungus in Native American sites further north. So it probably
there's fossil evidence for this way up north, yeah.
Yeah, so it's possible that this is, I mean, not to be alarmist, but it's the most difficult systemic fungus disease to control, but very few people who are exposed actually get a severe case.
One thing I want to go back to about global change and so on is probably the most rapid part of global change is the homogenization of the biota worldwide. And fungi have been really obvious in that we've been moving these pathogens across continental barriers. And so a lot of our big epidemics, both in ag and in forestry and in vertebrates like frogs and salamanders and so on, has been due to this movement of fungi across continental borders because of our rapid transit and all of our commerce and so on.
Yeah, absolutely. And we're going to get to populations that are susceptible, human populations that are impacted by fungi. But it's worth thinking about species that aren't humans that are susceptible. And it's just as Tom said, when we move a fungus from one continent to another and it encounters a host that it's never encountered, it either can't do anything and dies, or if it does infect it, it's like infecting a crop plant. There's a huge resource for the fungus of naive hosts, and it causes real trouble. And the frogs, well, the amphibians in general are the classic example, but there are others. Actually, plants are probably bigger.
Chestnut, elms, white pine, all the main forest pathogens that have been introduced, with a few exceptions, are fungal.
Yeah, I'm really interested in emergent zoonotic diseases, and especially in this time of COVID, when we've seen a SARS virus jump from presumably a bat host to humans, other lessons that can be learned from the fungal diseases on animals like bats experiencing white nose fungus, or the amphibians experiencing chytrid, and what we can do in terms of policy or education to maybe prevent future pandemics.
The first disease caused by the chytrid that killed frogs and, well, frogs, basically those amphibians, toads and frogs, that is now, that's been studied extensively by Tim James in Michigan and Matt Fisher at Imperial College London. And they have figured out that it came from Southeast Asia, and 30% of amphibian species have been seriously affected to the point of being exterminated, but not in Southeast Asia, because that's where this fungus was resident. And it evolved with the amphibians there, so it doesn't cause a severe disease. But in Central America, North America, Europe, etc., Australia, it's caused really severe disease. And there's another species of the same genus that is now affecting salamanders in Europe. And it's really damaged the populations in, say, Belgium, northern France. And the fear is it will get to the Mediterranean, where there's a large, huge distribution of these salamanders. In response, the US government actually got together in a bipartisan manner and passed the first law restricting the movement of fungi. And so it is aimed at restricting the international trade in salamanders to try to prevent bringing that disease to North America. And that's been very difficult to do for other industries.
Wow. And I just want to mention that I was in Tim James's lab. That's where I conducted my master's at the University of Michigan. And actually, so Tim, when I was there, was interested in the movement of bullfrogs for pregnancy tests. Bullfrogs were used before the urine tests, or were the first urine tests for pregnancy. And so they were moved throughout the world for that. But I got really interested in how fungi are moved in green unroasted coffee beans. So this morning when you had a cup of joe, there was presumably fungi in there that have been moved from Guatemala or Ethiopia or any of the other coffee growing regions into your home. Now, when you roast it and you brew it, you kill them, and they probably aren't metabolizing very much anyway, but we definitely move fungi across the globe through international trade.
Anytime you move biological material across zones, you're moving fungi. Any soil, any plant material, any animals, you're bringing in fungi with them.
Yeah.
There have been quite a number of articles in the popular press, like the New York Times, or especially the Guardian. And the BBC just had one by Roanna Hill. And there's a thing called the Millennium Seed Bank, where they bank these seeds from all over the world. And it turns out now they're using molecular biology to look at the DNA in these, and they find that not only do they have a Millennium Seed Bank, they have a Millennium Fungal Bank from all the fungi that are in these seeds. So it's kind of cool. Inadvertently, they're saving the genotypes of fungi.
Is that the one that's melting though? Like the structure is in the glacier and it's melting.
Oh no.
Okay, I would like to circle back to the conversation about valley fever, because I think, John, when I first met you and you were starting to delve into coccinioides, you were talking about prison populations that were suffering from valley fever, and how the cocci is in rodent holes that are near the jail. And I just want to highlight some of the ways that vulnerable populations are exposed to fungi. And that relates to my dissertation research, where I looked at mold in public housing. But can you share a little bit about the impact, social justice and environmental justice, and how it relates to fungi?
Well, the story on California prisons and valley fever is a tragic one. That they built prisons in Coalinga and Avenal, which are in a very western Kern County, maybe western Kings County. And that is the same area that's highly endemic for the fungus that causes valley fever. And so, you know, from a biological point of view, no surprise then, prisoners suffered disease, and so did the workers, the guards and the clerical workers at these prisons all have elevated caseloads, more than tenfold, I mean, huge elevation. And because prison population in California is overrepresented by Black and brown Californians, and there's evidence from a paper at publication from UC Berkeley by Leslie Louis that African Americans are more susceptible to the fungus genetically. And there's evidence from studies of prisons that Hispanic Americans are more susceptible. So what happened was the prisoners sued the state of California and said, you've put us in a place where more people, I think 40 people incarcerated in those two prisons died of valley fever over a period when one prisoner was executed in the state of California. So they correctly claimed that they were putting them in a very dangerous place. And so the federal government agreed with that. And the state of California then had to offer for every prisoner, would you like to get out of the Coalinga prison or the Avenal prison and be distributed in other prisons? And that's happened. But the source of the disease remains, the exposure to the disease remains, and that's what we're interested in now is looking at that. And we had the hypothesis that rodents carry this, wild native rodents like ground squirrels, kangaroo rats, pocket mice, carry this in their lungs without affecting the animal. And that then when they die though, the animal is adapted to use all the protein in that rodent. And the evidence that I heard at a meeting recently supported that idea, or in scientific lingo, did not disprove that hypothesis. So we're very interested in the relationship between the small mammal reservoir and the disease.
Mm-hmm.
But you should say something about your study of public housing and indoor air.
Well, Tom was one of the first to look at fungi in the indoor environment, right? With Anthony.
Well, with molecular methods, and certainly
Mm-hmm.
there was a long history of studying it before that.
That's true. That's true. Would you like to share some of what we've learned about fungi in the indoor environment?
Well, to begin with, I think what we learned was that a lot of what you breathe indoors is just a distillation of what you would breathe outdoors, and that in a normal healthy building, you're seeing fewer fungi than you would if you were outdoors, but it's the same mix. And your work in water damaged buildings was different, though, right? And there, what you see is that certain fungi are highly elevated indoors as a result, and so that's the main issue. And I would say that goes back to the pre-molecular methods that it's been known for a while that basically water in buildings is the problem.
Right. And then the exposure of those microbes in damp, moldy buildings causes asthma and respiratory disease. And so you have this compounded issue where people who are living in low income, federally subsidized housing, who have already called 30 times asking for renovations or leaking pipes to be fixed, get ignored, and then have these compounding health impacts from the water damage and then the mold that's there. So my PhD actually looked at public housing next door to privately owned apartments where there had been a hurricane, so in New York City after Hurricane Sandy, and we could see that even in all water damaged units, public housing had a more distinct microbial community than private housing that had also experienced water damage, which is, implicating neglect at the level of the housing infrastructure. So it's an environmental racism issue interacting with microbiology at this molecular level. So it's about 12:40 and we want to save time for Q&A from the audience. We've gotten a number of questions. So I'm going to share with you some of the audience questions. One is, are there any current initiatives or applications using AM fungi and ectomycorrhizal fungi to promote sustainable agriculture or forest restoration?
So for many years, I think that's been sort of the holy grail of mycorrhizal research, to be able to use these fungi to lessen the use of fertilizers, in particular in agricultural settings, and to facilitate growth of plants. And largely, though, I think the results of this have been, at least for an application point of view, have been disappointing, except from the money-making side of it, that people have put products out and sell them successfully. But I think really it's much more complicated than the science supports at the moment. That much of what happens when you add something to soil is that you're putting it into a very competitive arena where there's lots of other fungi already there. And so unless there's some way to make that particular fungus more competitive, it often doesn't evoke the result that you want. But in severely degraded ecosystems and so on, like strip mining and so on, adding fungi to those, mycorrhizal fungi to those, does have an impact. But it's a very small piece of this puzzle, I would say. So
with the agriculture, with the sorghum, we found there's a succession of these fungi, mycorrhizal fungi, and you would probably have to add the fungus that you want to add with the seed. You probably coat it with the seed, and there are a couple of fungi that are early in the plant, and then they're replaced later by sort of a dozen that seem to compete for the same niche. So I agree with Tom, it would be very difficult to add the fungus and have the timing and have it outperform the ones that are already there. Not impossible, but it's difficult.
And having stable inoculum, that is viable spores that you can store for the AM mycorrhizal fungi, the ones that would be associated with sorghum and a lot of the crop plants, is not trivial. They turn over relatively quickly, and so a shelf life of just a few months is what you're usually looking at. So it makes it quite, and they don't grow in culture, so it makes it quite expensive to generate the kind of inoculum you need, and then to get it to work in that setting is another whole set of problems.
But we now have the tools to actually try that. And I imagine people will. Somebody will try.
No, I think we can get there, but there's a lot of components that still need to be worked out.
That hasn't stopped companies from selling.
No, there's products on the market. There's
And
money
perhaps,
being made.
perhaps this is related to the next question is, can you talk about the prospects of agricultural biocontrol using fungi? I think this is kind of similar.
Biocontrol, actually, I mean, there are fungi that have been used successfully for biocontrol, especially of insects. But it's also sort of an emerging market with related difficulties in stockpiling what you need and having it stable and so on. But there have been more success stories on that realm. And biocontrol of introduced invasive plants is one area that fungi have been used in, although I think insect application in that has probably been greater usually, but there's been a few rust fungi, for example, that have been used in settings of invasive plants that have helped.
With the insects, is that entomopathogenic fungi that you're talking about?
No, I was talking about controlling invasive plants with insect predators of them. And so that's been done successfully in a number of cases.
There have been efforts to use fungi to control, for example, mosquitoes, and it hasn't been successful yet. The fungus that really kills mosquitoes has a very complicated life cycle. It has an alternate host. So it made it much harder to make it actually work.
Okay, we've got a question from Jillian Jacobson, a former undergrad student in the Brent Taylor lab. And they say, good to see you. And they've noticed that microbiome studies tend to focus solely on bacteria using 16S sequencing and ignore fungal species.
Well,
Do you have any suggestions for targeting both microbes in these type of studies? I did that.
I think actually there's a lot of studies where both get done. And I think the bias toward bacteria and archaea in them is simply because there's way more people that study bacteria and archaea than there are that study mycology. In fact, the number of major universities in the country that don't teach a mycology course is huge. We've been lucky at Berkeley to actually have that expertise for generations now, and be able to train other people. But there's a lot of schools where that training is not there. And so when somebody goes to study, some ecosystem or something, they'll go with the tools that they know, and they'll mostly study the bacteria. But I think really one of the things that's happened with the new tool sets of molecular diagnostics is that many more people that have not studied fungi before are starting to include them in their surveys and so on. So there is a bias, but it's much less than it was before those tools have come in. And certainly the tools are there to study fungi in the environment, and the number of studies that have been conducted with fungi is hard to even keep track of. There's so many now.
What we're hoping from the sorghum study is that the outcome, the fact that the earliest, largest, most coordinated response that the plant makes is to manage its mycorrhizal fungi, will cause other people to go, I'm not going to study this crop plant and not study the fungi, because the biggest story might involve the fungi. And at some point, we're hoping that if you want to publish something, you want to get a grant, you have to study the fungi along with the plant when you're going to do an ecological study.
Yeah. And I think the use of metagenomics and untargeted RNA-seq is helping to gain like a more holistic idea of microbial communities and all ecosystems.
Yeah, there's a professor at Berkeley, Joe Banfield, who is able now routinely to assemble the genomes of bacteria from environmental samples. And one of her students, Patrick West, has done it for a few fungi. And so that would be the future, that you could take any environment and get the entire genome of the microbes in that environment and all the genes that they're expressing. You'd know what's there and you'd know what they're doing. We're a ways from that for fungi.
And a part of the issue is the bioinformatics. That's kind of hard for the fungi. So I have another question that's about, that says, I know a histoplasmosis patient, but they have not been immunosuppressed, but they had it not due to immunosuppression. And it was acquired in an agricultural field. Wouldn't infection or pathogenicity also be dependent on dose at exposure? Maybe I should read that again.
Absolutely. So histoplasmosis and the valley fever, which is medically known as coccidioidomycosis and related diseases, can affect otherwise healthy individuals. And if 1,000 people got histoplasmosis or valley fever, maybe only 40% would feel it, would know that they'd been infected. And maybe only 10% would have a case that if it wasn't treated, it would kill them. And so, yeah, in fact, I am valley fever positive. I have living fungus in my lungs, and my immune system has walled it off in a granuloma, no problem, which is what it does with those little rodents. So it infects them, but it doesn't hurt the rodent. Then when the rodent dies, it's a food source for the fungus.
But the other part of that question was the role of inoculum level.
Oh yeah.
Like when you're breathing in a whole bunch of spores, it's a different game, right? And
absolutely.
Probably even in cocci, it's when people, there's a big dust storm or something, and you get a lot of spores in, and you haven't seen that before, that you end up in serious trouble.
That is absolutely correct. And when we know that is when they call it an outbreak, when 7 or 10 people get sick all at once.
Right.
Almost invariably, they were excavating soil in the area where the fungus is known to be. So they breathed in a very large number of spores. And that's probably what happened to the person who has histoplasmosis.
Yeah, histo is usually birds, right? That like
birds and bat guano.
Yeah.
It's different than cocci, but not in a lot of ways. Yeah.
Another question that I can say yes to already, but I'll pass to maybe Tom is sudden oak death caused by a fungus?
Depends on your definition of a fungus. The way we talk about fungus in class is fungi with a big F being the fungi that are in the kingdom fungi, and fungi with a little f being the fungi in several different kingdoms, the organisms in several different kingdoms that behave and look like fungi. And so the one that causes sudden oak death is one of those. It's an Mycota that behaves and looks like true fungi, but is not actually a true fungus. But it tends to get studied by people like
mycologists
study fungi.
Yeah, and I like to say the queendom fungi, just to kind of challenge the gender hierarchy in our scientific language. There's another question that I think is really interesting coming from the Innovative Genomics Institute is, are people using CRISPR to edit fungi?
Yeah, we need Louise Glass here. But,
people have gotten CRISPR to work in fungi.
Yeah.
It's not as wonderful as it would be if fungi made a big cell, because you can inject animal cells and big plant cells, but for fungi, you still have to transform the fungus to get the CRISPR machinery in there. And so it hasn't been quite as easy to use as it would be if they just made bigger cells. But yeah, it works. Actually, Rachel Brehm too would be another one to. Two mycologists, Rachel Brehm and Louise Glass, members who study the queendom fungi. They both know about how to use CRISPR on fungi.
Cool. And someone is wondering about fungal evolution. How fast do fungi evolve, and how do you study fossilized fungi? Can you sequence remnants?
Ooh, on the how fast, I want to jump in there about this paper that came out last year or the year before on the mutation rate in the humongous fungus, the honey mushroom, and just say that has an extraordinarily low mutation rate, like orders of magnitude lower than what's observed in other organisms. And there may be some artifact involved there, but at least the observable accumulation of mutations in that fungus is extremely low. So you often think of fungi as being things that turn over very quickly, but I think it's mostly because the population sizes are so high that you're looking at lots of accumulated mutations over the whole area. But other than that, they're typical eukaryotes and have what we think are very, very similar mutation rates to other organisms.
Yeah, if you apply a strong selection, for example, the azole antifungals on Aspergillus Fimigatus, you evolve resistant ones in a hurry. And so it looks like they've been evolved independently in different agricultural areas around the world.
And again, that'd be one with a huge population size, right?
Very, very large population size and very strong selection. You know, either you reproduce or you don't. And so then any mutation that gives you an advantage there sweeps through the population very rapidly. So fungi evolve just like anything else. Some may have a very slow mutation rate, but they get there.
And maybe one difference would be that many fungi have a mitotic phase that can last, for decades or millennia even. And so it has both the option of sticking with a particularly successful genotype for long periods of time, and the option for recombination. And that plasticity, I think, is really useful for being successful over long term for these organisms.
Yeah, humans are trying to clone humans, but fungi have been doing it no problem.
Yeah, they clone.
Another question is, would you mind sharing your opinions regarding the use of psilocybin in modern mental health treatments?
I'm for it. I think the available evidence is pretty compelling that it has some real utility, and I'm very happy to see that it's once again getting investigated, to determine how useful it really is and how to use it. The fact that it's been unstudied now for decades because of the laws that came in the early 70s has been very unfortunate. I think it's a potentially an extremely useful pharmaceutical.
Yeah, I support that same idea, and note that we know that fungi make compounds that can really modify animal behavior. And we know it from insects, the zombie ants and the flies, that when they get a fungal infection, totally changes their behavior to the benefit of the fungus. But so fungi are clearly making compounds that have specific effects on animal nervous systems. Very useful.
Yeah, and something we didn't get to earlier, but I think is relevant now, is I'd like to talk about the use of fungi, including psilocybe, in cultural practice and shamanism and mythology. So we know in Central America, psilocybe has been used for religious ceremonies for hundreds of years.
Thousands, yeah. Yeah.
Yeah, so it is actually approaching 1 o'clock. So that is our time. I think I want to take a moment to thank our panelists, John Taylor, Tom Bruns, and also Lou Feldman, who introduced us, and say happy anniversary, P&B.
And thanks to everybody who came on and listened to this during their lunch hour.
Yeah. And it was also great to see you, Iman and Tom. And I'm hoping very soon we can see each other in person. And Lou.
Maybe in the gardens. All right, thank you all.
Bye-bye.
Bye-bye.